Good morning students, welcome back to online class. Today we are going to do the second part of biomolecules. Last time we have studied about the carbohydrates, proteins, nucleotides, then different parts of the saccharides, that is monosaccharides, oligosaccharides and the polysaccharides. So here we are going to study about the glucose, how glucose is prepared. So in the preparation of a glucose, you know the formula of glucose, that is C6H12O6. So how it is prepared? So it is prepared from the sucrose. So the sucrose, how you will get? You get from the sugar cane. So sucrose is boiled with the HCl or H2SO4 in an alcoholic solution. So we will get glucose and fructose. So C12H22O11 plus water with the dilute HCl, right? It's dilute, very dilute, or H2SO4, it gives glucose and fructose. And the second formation of the glucose is from cellulose, that is from starch or cellulose, you know, both are same. So C6H10O5 number of times plus water, it gives at a certain temperature and the atmosphere pressure, it gives number of glucose, that is C6H12O6. So this is a formation of preparation of a glucose from sucrose and starch. So the formula for the glucose is what? Glucose means aldehyde and alcohol will be there. So CHO, CH, OH 4 times and then CH2OH. So glucose is aldohexose because aldehyde is there, and is also known as a dextrose. So glucose means dextrose. So it is the monomer of many of the large carbohydrates, so namely starch and cellulose. So these are all the importance of the glucose. That is molecular formula is was found to be C6H12O6. So from the structure glucose, it gives N-hexane when you apply heating at the temperature with the hydrogen iodide, it gives N-hexane. The second one is what? From the glucose, when it reacts, it gives, in the presence of NH2OH, that is hydroxylamine, it forms oxime and adds a molecule of a hydrogen cyanide to give cyanohydrin, CN, right? So it gives what? Ketone group. And finally what it gives? It gives the cyanide and the ketone group in the glucose form. So glucose, when reacts with the hydroxylamine, it forms the oxime and adds a molecule of a hydrogen cyanide to give a cyanohydrin. So these reactions confirms the presence of carbonyl group. Okay, because cyanide CN is there, so it gives a cyano group. The next one is what? Glucose, when gets oxidized, it gives a 6-carbon carboxylic acid. How? Like gluconic acid. How it goes from aldehyde to carboxylic acid in the presence of Br2 bromine water. So the 6th term, it gives CHO to COOH. So this is glucose to gluconic acid. The next one is what? Acetylation. So with the acetylation process, the glucose, it gives what acetic, with the presence of acetic anhydride, it gives glucose pentacetate. See here, acetic anhydride means what formula? CH3CO twice O, right? Acetic anhydride, CH3CaOO twice O, right? So it gives what? Pentacetate. How it gives? Aldehyde will be same. Over here with this O, it gives COCH3 with the 4 times, and here also COCH3 will be there. Okay, so it gives what is the name of this one? Pentacetate, glucose pentacetate. Okay, this is the process of the glucose. So from the glucose with HI, and then hydroxylamine, that is NH2OH, and with the bromination and the acetylation, that is the acetic anhydride. So the configuration of a glucose, configuration means what? The structural formula of the form of the glucose. So glucose means C6H12O6, so aldehyde and OH is there. So scientifically, scientist Emil Fischer has determined the configuration of the glucose in a form of almost aldopentose and aldohexose. So the scientists have discussed this one, as given the term D, L, small d or plus sign. right? L or minus sign for the term glucose and fructose. So D and plus sign plus gives minus minus no. D hyphen plus hyphen glucose. The name is given. Why the name is given? See here in the Fischer projection carbohydrate compound, if the OH group is attached to the bottom, OH group is attached to the bottom of the most chiral carbon atom on the right side. This is the right hand side, this is the left hand side. So OH group is on the right side, so the compound possesses a D configuration. So if OH is attached on the left side, it gives L configuration. So you can mark this one, right? So OH on the right side, it gives D configuration. OH on the left side, L. That's why L is there. Left side, it's a L configuration. All right. So DL and L compounds are enantiomers. So optically active organic compound rotates in the plane of polarized light. So it rotates. So then from that we can give the name also. And basically the down one only we have to remember, focus on that. So if only optical active organic compound shows the rotation, that is a clockwise, that is on the right side direction, then it gives plus. When it shows the clockwise operation and that is also on the right side, so the name is D rotatory, that is a D of dextro rotatory. So it is mentioned that D or plus, if optically active organic compound shows the rotation in the anti-clockwise direction, that is on the left side, it's a negative sign and the name is come levorotatory. Understood? So the OH is on the right side, so it's a D, capital D or plus sign, right? And the OH is on the left side, so the name is L or negative sign. Okay, so it's a dextro for the right and the L, levorotatory for the left, L left, right? So this is the structure of the configuration of the glucose. See here, this one is on the right side, so D plus glucose. OH is on the right, left side, so it's a L, levorotatory, anti-clockwise, so it's a L and minus glucose. The name is same. Okay. The cyclic structure of glucose, the aldehyde group which is present in the configuration of a glucose, but glucose does not give some reaction of aldehyde. So which is they are not given by the aldehyde is what? Glucose does not form an addition product with sodium bisulfite, that is NaHSO3. Generally, aldehyde group contains compound form addition product with the sodium bisulfite. So the tolerance reagent we are going to study in the 12th standard in the functional group. So an aldehyde reduces tolerance reagent and the Herring solution, it gives a violet color. The shift reagent, so glucose gives first two reaction but does not give violet color with the shift reagent. So this, when you do the practically, then you will get color change properly. So you will understand that. And aldehyde does not show mutarotation property, while glucose shows this property. So there is a difference between the aldehyde and the glucose. So see here, the rotatory is what over here. See the two cyclic formation of in the equilibrium position. The glucose one, see it is going on this direction. So alpha D and then plus glucose, and it rotates. It gives from alcohol to aldehyde, and again it gives the term this one. So it's a D plus glucose. So it is rotating two cyclic hemi-acetal form of a glucose which differs only in the configuration of the hydroxyl group. It is called anomeric carbon. So what is the name of this one? Anomeric carbon. So you can say two cyclic form of a form which exists in the equilibrium position. Now the next one is a fructose. You can see the diagram. Fructose means ketone group is there, ketohexose is there. So with the same formula C6H12O6, the structure is there, but the thing is what, only ketone group is there. So ketose, or we can say ketohexose, right? So the last term is on the right side, OH is on the right side, clockwise rotation is there. So the name is D minus fructose, that is a ketone group, okay. So D minus fructose by the addition of alcohol at a C5, which gives a ring form. It's a 5-membered ring. So we can say give a name of furan, 5 number furan. If a 5-membered cyclic compound with oxygen forms 4 carbon atom. We have studied in the 11th standard about the fructose furon, having a 5 member with a 4 carbon and 1 oxygen. Okay, so here structure of a furon, 4 carbon with a double bond and 1 oxygen. Furon, it gives to fructofuron. See here 1, 2, 3, 4, 5, 6. Okay, fructofuron, which shows 1 ketone group over here, and it gives to the alpha and the beta formation of a cyclic structure of a 2 anomers of the fructose. So basically you can write in this form also, both are same only. Fructofuranose, you can write the structure same. How you will write? Furane and then CH2OH on the both the side and HNOH on the opposite. You have to find this one. Now about the protein. We are going to study about the protein. So protein formation is what? With the CONH group. CONH means amide group, which is present in the structure. Then there is a protein formation is there. So the most abundant organic molecule of the living system, they forms about 50% of the dry weight of the cell. Protein is required in our body. So the formation of the protein is what? Protein on complete hydrolysis gives amino acids. So there are 20 standard amino acids which are repeatedly found in the structure of a protein, that is in animal, plant or microbial. So the protein synthesis is controlled by the deoxyribonucleic acids. Okay. So amino acids means what? See here an amino group, that is the NH2 group, amino group, NH2 group, CHR, R means alkyl group, and COOH, carboxylic acid group. So amino acids are a group of organic compound having a 2 functional group, that is what? Amino and the carboxylic acid. So we have to attach this one with the other terms so that you will get the protein properly. So first we will study about the amino acid structure. So NH2 group is a basic, whereas carboxylic acid is acidic. Amino is a basic, and carboxylic acid is acidic. So R can be H in a glycine, CH3 in alanine, hydroxy methyl in serine. In others, it can be a hydrocarbon chain. So all amino acids contain carbon, hydrogen, oxygen, and nitrogen, but some of them additionally contain the sulfur also. So amino acids can be differentiated into the 7 groups, like protein. See here, protein are a heteropolymer of amino acids. So there are polypeptide link is there, having the same chain. See here, amino acids NH2COOH, amino acids NH2COOH. So when these two combine, it forms amide link. See here how. So this hydrogen and OH form a water. So OH, then CO, then RCH, NH, and then CO. So NH here, I'll take another color so you will understand properly. So NH and CO form what? Amide link. NH and CO forms what? Amide link. So this one is the amino acids. Amino acids form NHCO. So it's a protein structure. So see here, CONH is a peptide bond. Other one is same only. Okay. Till here we are doing. Thank you. Hope you understood topic very well. Next time we will discuss about this one in deeply. Thank you.